Okay, welcome back to the Science Shack, where the next video in our respiration series. It's going to be a very quick recap of what you should have done already. Maybe you already learnt this in class. This is a video on the link reaction and the Krebs cycle, covering these bits of syllabus right here. So we're now going inside the mitochondria, and we're going to look at what happens from pyruvate right the way around this really important central cycle of metabolism called the Krebs cycle. So moving on very swiftly to a quick recap. You should have already watched the first video in this series and know that this is called glycolysis. Okay, so glycolysis involves the phosphorylation of glucose to glucose phosphate. It then breaks down to two triose phosphates. They then get oxidized to pyruvate, leading to reduced NAD and the production of some ATP. Now, you know, I always like to make this a multi-tiered sort of difficulty thing. The one on the left is absolutely fine for A-level purposes, but I would say that is kind of the bare minimum. There's actually a lot more going on in there. Okay, so you can see some of the other side reactions kicking in here, which we'll come to a little bit later. So you've got the formation of glycogen, pentose and nucleotides from that glucose phosphate, and where fats come in. You should all know we can use fats in metabolism. That triose phosphate can be easily formed from glycerol, and then that gets turned into pyruvate, and you can see that we're entering at the very bottom of that diagram, we're entering into the mitochondria. This little bit here, see anaerobic conditions. Now, depending on what organism you're talking about, you're either going to produce lactate or ethanol, but another video on that coming in the future. Okay, so that's what we've just done. Let's get straight into it then. So once inside the mitochondria, the first thing that happens is a thing called the link reaction. And this basically is a short little section that's a go-between between the pyruvate and entering the Krebs cycle. So what happens is we end up with another oxidation reaction. So pyruvate is oxidized to acetate by removing the hydrogen and obviously the electrons as well. That's what makes it reduction. Hydrogen is used to make more of this reduced NAD, which we made in glycolysis. Now this is how these high energy electrons and these protons are going to get transported to oxidative phosphorylation, which is the last section. Acetate, obviously very unstable. Right, we want unstable molecules at this point, so we can actually generate some release some energy in that really controlled way we talked about in the last video. So once you've got that acetate, you need something to temporarily stabilize it, and this is where a coenzyme comes in. We've got acetyl coenzyme A at this point. So coenzyme A comes and binds to the acetate, makes acetyl coenzyme A, and that then transports our acetate into the Krebs cycle. That's all there is to the link reaction, but up in the title there you can see there's actually two versions of this per glucose, right? Because obviously we split the glucose in half to make the triose phosphate, okay? And so we need to think about this in terms of doubling it up. So for every glucose's worth of link reaction, we make 2 reduced NADs and we make 2 carbon dioxides. Okay, so be careful if you're ever asked a question on this as whether they're talking about per pyruvate or per glucose. Right, so on to the Krebs cycle. So this is named after an amazing British biochemist called Hans Krebs. Right, he spent his life's work making this incredibly complicated, incredibly detailed, incredibly important discovery, and I'm going to give you a really stripped down version of it. Okay, so that's Hans Krebs there. Okay, and that down in the bottom right is the version I'm going to show you now. Because for all, if you just want to get through the A level and that's all you're looking for, all you need to know about the Krebs cycle is on this slide. I am going to give you obviously the next tier up. I'm not going to give you the most advanced. I'm going to save all of that for a separate video at the end of this series. Okay, so this happens in the mitochondrial matrix. What happens, as you can see, our acetyl coenzyme A here, right, brings that acetate in and combines it with a 4-carbon compound. Now remember, the whole point of respiration is we're releasing energy gradually in lots of little stages, gradual. Okay, so acetyl CoA combines the acetate with the 4-carbon compound to make a 6-carbon compound. Now this cycle is also called the citric acid cycle because that 6-carbon compound is called citrate. Okay, so we're not going to go into the names just yet. I've got to hold myself back to do this really basic version before we can go any further. Okay, This 6-carbon compound then very gradually gets broken down into a 6-carbon, 5-carbon, and back into the 4-carbon compound. That then regenerates this 4-carbon compound so it can go round again. As a result of that, you break off little carbons which form carbon dioxides, and you also reduce more coenzymes. So in each turn, you reduce 3 NADs, so that's 6 per glucose, because it's doubled up with 2 acetates per glucose. 1 of these FADs or FAD2s, so 1 of those, so that's 2 in total, and you make 1 ATP per turn. Okay, technically it's GTP, but it doesn't matter. You make ATP by substrate level phosphorylation. Okay, so 2 in total. And that goes round and round and round again. Now, if you've been counting carefully, as we're going to come to at the end, that actually accounts for all the carbon dioxide in your GCSE understanding of respiration. That's all 6 accounted for. Okay, so acetyl coenzyme A reacts with the 4-carbon molecules. This is the syllabus, releasing coenzyme A, producing a 6-carbon molecule that enters the Krebs cycle. In a series of oxidation and reduction reactions, the Krebs cycle generates reduced coenzymes and ATP by substrate level phosphorylation, and carbon dioxide is lost. So this really simple version here is enough, but I think we can do better than that. Okay, I prefer something that looks a little bit like this. Okay, so there's our link reaction. Pyruvate, the 3-carbon molecule, breaking down, being broken down, carbon dioxide breaks off. We produce one of these reduced reduced NADs or NADH here. Just the hydrogen is shown in this diagram. Acetyl coenzyme A picks up the acetate, takes it into the Krebs cycle. Now there are a lot more chemicals in here, but these are the sort of big players. The 4-carbon chemical, right, that's combined with the acetate is called oxaloacetate. Okay, that combines with the acetate to make citrate. Okay, that's our 6-carbon molecule. So sometimes you see this called the citric acid cycle. Citric acid cycle, Krebs cycle, same thing. That confused the hell out of me for months in my first year of my degree. So if you're going to go that route, then please don't be similarly confused. So then, as we break this down, we're removing carbon dioxide. That hydrogen comes off there to reduce an NAD to NADH. We form our alpha-ketoglutarate, which is a fantastic word for a 5-carbon molecule, alpha-ketoglutarate. Then we get our 2 NADs and 1 FAD over here, these 3 hydrogens, that one. Carbon dioxide breaks off, and it's in this stage between the alpha-ketoglutarate and the oxaloacetate that we generate our ATP by substrate level phosphorylation. Okay, so that's your basic. That there is a little bit more. Okay, just a little bit sort of intermediate level, if you like. So if we put together everything that we've done in respiration so far, what we get this. Okay, so here's our glycolysis. This is all happening in the cytoplasm. Right, there are all our stages. Pyruvate imported into the mitochondrial matrix. Right, the link reaction generates acetate, which combines with acetyl coenzyme A, moves round the Krebs cycle here, breaking down very, very, very gradually, releasing those hydrogen ions, releasing those electrons to reduce those coenzymes, which are then going to be used in the next stage, which is where we get the payoff, where we get all the ATP being produced, and that is the electron transport chain. Okay, so coenzymes massively important, and an essay question you might get on the end of paper 3, if you're doing the same syllabus we are, is coenzymes are important in biology. Describe how, 25 marks. Sounds complicated, but of course, actually, when you think about it, there's an awful lot you can talk about. Okay, what coenzymes are is they're molecules that bind with a specific enzyme or a substrate and help to catalyze reaction. Now they must be regenerated. Okay, there's not an infinite supply. They have to be regenerated, and that fact becomes really important when we discuss anaerobic respiration. Put 2 examples down there. That's your NAD there, and that's your FAD. Okay, they stand for nicotinamide adenine dinucleotide and flavine adenine dinucleotide. Coenzyme A, right, is another coenzyme that you will have seen in this process. Apologies if you can hear the science children outside the science shack. It has just become feeding time. So, NAD, FAD, and coenzyme A can all accept hydrogens. I like to think of them as basically wheelbarrows that drag these other molecules around the different sort of parts of respiration. They're all used to form a proton gradient that makes ATP from ADP and PI in the electron transport chain. Okay, coenzyme A obviously is there to do the transition between glycolysis and the Krebs cycle in the form of the link reaction, and that's a picture of coenzyme A at the bottom there. Okay, so you do need to know that they're important and they do need to be regenerated. So, what's a coenzyme? Well, it's a molecule which helps an enzyme to function. What for do you have to know? NAD, FAD, going back a bit, NADP, and hydrogen carriers, and coenzyme A helps bring acetate into the Krebs cycle. How do you remember which one's in photosynthesis? Well, it's the only one with a P in it. That's not what the P stands for, but that is the truth. Okay? So, to conclude, before the science children outside get too noisy, right, what we've got here is we've got used up so far in total, all the way down the side here, we've used up 1 glucose and we've put in 2 ATPs. Okay? Out of that, we've generated 6 carbon dioxides. That's all the 6 carbon dioxides in your GCSE understanding of respiration. 10 NADH and 2 FADH2 per glucose. We've also generated 6 ATP by substrate level phosphorylation. And it's in the next stage, right, where we see the payoff for these coenzymes. Many thanks, and I'll see you then.